Rod cGMP-specific 3 feet. 5 cyclic phosphodiesterase subunit beta is the beta subunit of the protein complex PDE6 that is encoded by the PDE6B gene. PDE6 is crucial in transmission and amplification of visual signal. The existence of this beta subunit is essential for normal PDE6 functioning. Mutations in this subunit are responsible for retinal degeneration such as retinitis pigmentosa or congenital stationary eye blindness. Structure: PDE6 is a protein complex located on the photoreceptor's outer segment and plays an important role in the phototransduction cascade. There are two types of photoreceptors: cones and rods. The rod and cone PDE6 complexes have different structures. PDE6 beta together with PDE6 alpha and two identical inhibitory subunits, PDE6 gamma, form the rod PDE6 holoenzyme, while the cone PDE6 complex only consists of two identical PDE6 alpha catalytic subunits. PDE6 beta, one of the catalytic units in rod PDE6, is composed of three domains: two N-terminal GAF domains and one C-terminal catalytic domain. The non-catalytic GAF domains are responsible for cGMP binding. The C-terminal interacts with cell membrane by isoprenylation and S-carboxyl methylation. Function: absorption of photons by rhodopsin triggers a signal transduction cascade in rod photoreceptors. This phototransduction cascade leads to hydrolysis of cGMP by cGMP phosphodiesterase (PDE) that closes cGMP-gated channels and hyperpolarizes the cell. PDE6 beta is necessary for the formation of a functional phosphodiesterase holoenzyme. Function of PDE6. PDE6 is a highly concentrated protein in retinal photoreceptors. With the presence of the GAF domain, PDE6 can actively bind to the cGMP. The inactive PDE6 in the dark allows cGMP to bind to cGMP-gated ion channels. The channel remains open as long as cGMP is binding to it, which allows constant electron flow into the photoreceptor cell through the plasma membrane. Light causes the visual pigment rhodopsin to activate. This process leads to the release of subunit PDE6 gamma from PDE6 alpha beta, activating PDE6, which leads to the hydrolysis of cGMP. Without the cGMP binding, the ion channel closes, leading to the hyperpolarization. After hyperpolarization, the presynaptic transmitter is reduced. Next, the enzyme guanylate cyclase restores cGMP, which reopens the membrane channels. This process is called light adaptation. Function of PDE6B. PDE6 beta is the only protein that undergoes the two types of post-translational modification: prenylation and carboxymethylation. The geranyl group of PDE6B is the result of these modifications, which are responsible for the rod PDE6's interaction with membrane. Animal studies. Rod1 mouse. Mutation of the PDE6B gene leads to the dysfunction of PDE, which results in failure of hydrolysis of cGMP. The Rod1 mouse is a well-characterized animal model of retinitis pigmentosa caused by the mutation of PDE6B gene. The phenotype was first discovered in rodless mice in the 1920s by Keeler. An insertion of murine leukemia pravirus is present near the first exon combined with a point mutation, which introduces a stop codon in exon 7. In addition to the Rod1 mouse, a missense mutation, our 560C, In exon 13 of the PDE6B gene is the character of another animal model of recessive retinal degeneration. In ROD1 animals, the retinal rod photoreceptor cells begin degenerating at about postnatal day 10. And by 3 weeks no rod photoreceptors remain. Degeneration is preceded by accumulation of cGMP in the retina and is correlated with deficient activity of the rod photoreceptor cGMP phosphodiesterase. Cone photoreceptors undergo a slower degeneration over the course of a year, which causes the mutants to completely go blind. The possibility of altering the course of retinal degeneration through subretinal injection of recombinant replication defective adenovirus that contained the murine cDNA for wild-type PDE6B was tested in ROD1 mice. Subretinal injection of ROD1 mice was carried out 4 days after birth, before the onset of rod photoreceptor degeneration. Following therapy, PDE6B transcripts and enzyme activity were detected, and histologic studies revealed that photoreceptor cell death was significantly retarded. The albino FVB mouse laboratory strain became blind by weaning age due to a mutant allele of the PDE6B gene. There are pigmented derivative strains of FVB that lack this trait. RCD1 dog. Similar to ROD1 in mice. Rod cone dysplasia type 1. RCD1-PRA is a form of progressive retinal atrophy, PRA, with early onset of the disease. The Irish setter is a characterized animal model of RCD1. The mutation is caused by a nonsense mutation in PDE6B gene. Photoreceptors start degeneration at postnatal day 13 until a year after the dog is totally blind. Further reading. Jenner Reviews, NCBI, NIH, UW entry on retinitis pigmentosa overview.